All right, time to go teach. Hello and welcome to our final episode on our unit of biochemistry. So let's look at nucleic acids. Let's get this thing started. Our first learning target of the day is to compare and contrast the two types of our nucleic acids. So clearly there are two types of nucleic acids and we're going to compare them. Next, identify the monomers that make up nucleic acids. So we're going to have to be able to identify the different monomer that makes up these specific nucleic acids. And lastly, describing those specific monomers. What makes these monomers special? How are they set up? What's going on with that? So let's get this thing started. Our nucleic acids, there are two types, RNA and DNA. RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. And we will look to see why in the world we have these two different types of nucleic acids. And the monomers for both of them are called nucleotides. A nucleotide is made up of three different parts. It is made up of a phosphate group. Now, a phosphate group we typically think about as being a phosphate in the center and 4 oxygens around it. We could also draw it with some hydroxyls, so we could add our hydrogens as well, but that is generally the structure for a phosphate group. Then we have a carbohydrate. This specific carbohydrate, that is ribose. We'll look at what makes ribose, well, what makes it being ribose, and what makes deoxyribose different from it. And lastly, it's made up of a nitrogenous base. And so we can see all the different nitrogen atoms here. And they're also called nucleobases. There are 5 different nucleobases out there. It's adenine, thymine, guanine, cytosine, and uracil. So those are the 5 different nucleobases we can have. So let's check this out. First off, what makes our carbohydrate ribose or deoxyribose? Well, remember how to count those carbons? So this is our first carbon, and this one, that's our second carbon. And that second carbon is going to be really important for knowing if it's ribose or if it's deoxyribose. This happens to be ribose, and we know it's ribose because of this oxygen right here. So this molecule is ribose. Deoxyribose means that we are removing an oxygen. To deoxy means to take away an oxygen. So deoxyribose would be like this, where now we have taken away that oxygen upon that second carbon. And so because our second carbon is missing an oxygen being attached, it is deoxied. It is a deoxyribose. The nitrogenous bases. This first one is adenine. The second one right here, this would be called guanine. These are specific nitrogenous bases that are called purines. We know that they're purines because they have a double ring structure. So this double ring structure is called a purine. On the other hand, the other 3 nitrogenous bases, of which thymine is one, and then we could have cytosine right here. And the last one, uracil, you'll notice that each of these are only single ring structures, and these are all called pyrimidines. So uracil, thymine, and cytosine, those are pyrimidines, and adenine and guanine, those are both called purines. Adenine, guanine, and cytosine are found in both RNA and DNA. Thymine is only found in DNA, and uracil is only found in RNA. And when we're building a nucleotide, so in this case, we have our ribose sugar. We're going to add a phosphate group to the top. So a phosphate group will be attached to the 5th carbon, and our nucleobase is going to become attached to the 1st carbon. So this is a nucleotide. We have our phosphate group, we have our sugar, and we have our nucleobase. There we go. Those are the 3 parts that make up our nucleotide. The phosphate group, the carbohydrate, and the nucleobase, or the nitrogenous base. In this case, it's adenine. And a nucleic acid, well, they come together just like every other biomacromolecule comes together. It's going to use dehydration synthesis. So we bring our other nucleotide in, and we are going to have dehydration synthesis occur. And there we go. Now we have two nucleotides bonded together, and we should be able to recognize where the different carbons are. The fifth carbon of our sugar is facing right here. On the bottom, you can see that our third carbon is facing downward, and it always has this orientation. So the third carbon is going to be facing in one direction, the fifth carbon connected to our phosphate group, which is then connected to the third carbon of our next nucleotide. So they always, each of our nucleotides have the exact same orientation. The third carbon is facing in the same direction as the third carbon of the other nucleotide. The fifth carbons are both facing in the same direction as each other. So our nucleotides always following a very specific way or a very specific orientation. And that means that we would identify them as having ends. This is the 5' end, and this is the 3' end, based upon where the third carbon is facing and the direction that the fifth carbon is facing. That is how we identify the directionality of our nucleic acid. And in this case, this is RNA, and we know it's RNA because right there, we have an oxygen upon the actual second carbon. And so since we have that oxygen, that's ribose, and that makes this a strand of RNA. RNA is a single-stranded molecule. On the other hand, if I were to change things up a little bit, now we have DNA here because we just removed the oxygen upon our second carbon. And we're going to change the way that this looks a little bit. I'm going to use some other symbols. So now my carbohydrate is going to be just like this. Now this is going to be the symbol that I use for my phosphate group. And I'm going to change my different nitrogenous bases to use different symbols just to make life a lot easier to see and represent how DNA is going to come together. Because DNA comes together in a double-stranded way. Unlike RNA, DNA will be double-stranded, based upon hydrogen bonding that exists between the different nitrogenous bases. So adenine is able to hydrogen bond to thymine, and then you have two hydrogen bonds that hold them together. You will also notice that the orientation of both of our strands is in the opposite directions. We describe this as the strands being anti-parallel because they are parallel to each other but facing in the opposite direction. The 5 carbon right here is facing downward. The 3 carbon up here is facing upward, which is the opposite of this other strand where the third carbon is facing downward and the 5th carbon is facing upward. And so, because of that opposite direction, we call it anti-parallel. If I were to add some other nucleotides, you can see that guanine, well, it hydrogen bonds with cytosine. Cytosine and guanine don't have 2 hydrogen bonds, they actually have 3. So they have a stronger force of attraction between them than the adenine and thymine do. But adenine and thymine always pair with each other. Guanine and cytosine always pair with each other every single time. And our DNA is going to have that anti-parallel nature to it. And that's going to be important later on when we're looking at genetics. But we won't go into the specifics until then. So in summary, there are only 2 types of nucleic acids. It's the RNA and DNA, and that's based purely upon that sugar that happens to be in the middle of our nucleotide. Is it ribose or is it deoxyribose? That determines which type of nucleic acid we have. Every single nucleic acid, though, is made up of nucleotides, which have those 3 parts: the phosphate group, the carbohydrate, and the nitrogenous base, of which there are 5 different nitrogenous bases: adenine, guanine, and cytosine being found both in RNA and in DNA, thymine only being found in DNA, and uracil only found in RNA. So that is the way that we can think about those. The nucleotides are always going to bond together using dehydration synthesis, just like every other biomacromolecule that we've gone through in this unit. So every single time that you're going to take those different monomers and put them together, remember It's going to remove the components of water so those different monomers can bond together. All right? And that's it for this time. Be awesome. Stay awesome.